To this revision video for IB Biology, and today we'll be looking at DNA replication. You need to understand that DNA is a stable nucleotide for storage of information. So the basic structure of DNA can be seen here, that we have this anti-parallel backbone of phospholipid, phosphate and sugars, and the sugar is obviously deoxyribose, a pentose sugar, which you should already really know. Between the bases there are hydrogen bonds. There are more hydrogen bonds between G and C, and overall there are generally more G and C bonds in DNA. It needs to be stable because otherwise you will have a lot more copying errors, and that is obviously an issue. So it is important because it is the base sequences which code for amino acids and then for proteins. The molecule is extremely long, so it can have lots of information. Base pair rules means that you can copy and replicate the DNA. The double helix structure gives it more stability, and the hydrogen bonds give it stability, but they're easy to unzip to allow replication. So when we're talking about DNA replication, we're actually talking about interphase, which occurs before mitosis, and that is where the DNA is replicated. And the replication of DNA means you get an exact copy with the same bases in each strand. And there are two purposes for DNA replication. One is for reproduction, and it might be used for the egg and sperm. Obviously that is through meiosis, and there's extra division there, but also for mitosis, for asexual reproduction. And the other purpose is for growth and repair. If we need to make genetically identical daughter cells, we need all the DNA to go forward. So this shows you the basic stages of the DNA replication. So here we have a proportion of DNA about to undergo replication. The first thing that happens, and there is actually another stage here where my pen is now called DNA gyrase, which actually twists the DNA. So in terms of spelling that, the enzyme is called gyrase. Okay, but then once the DNA gyrase has happened, then we go on to here. And we go on to this bit here where the DNA helicase is the enzyme that unzips the hydrogen bonds or breaks the hydrogen bonds between the DNA molecule. That then leaves us with two separate strands, and three nucleotides which are made within the nucleus are then able to combine with their complementary base pairs, and they form two strands as a result. So we have two new molecules of DNA, one with the original strand there, and then this one with a new strand, and that is called semi-conservative replication. An enzyme then links these strands together, so you can see this nucleotide here is not attached to this one at the moment, which is not attached to this one, which is not attached to this one, and DNA polymerase binds those together, and we'll talk about that in more detail for the higher level students shortly. And said this method is called semi-conservative replication, because if you look here, this was the original strand, still the original strand here, that's your original strand, that's your original strand, whereas this one here therefore is a new strand, then that's made out of these bits here to make the new strand. So one strand in the new DNA molecule is an original strand, one strand is old. Sorry, it's a new strand, and this process is called semi-conservative replication. So there are a couple of different theories out there originally. One might be that it is conservative replication, and you start off with one parent strand, you end up with the original strand, and then one new strand. But that has been proven wrong, as we'll talk about in a minute. We now know that it's semi-conservative. So from the original strand, you end up with the original molecule of DNA, you end up with one old strand and one new strand. There is also another model called the dispersive model, which you can look at if you would like to. How do we know this? Well, we know this because of the work of two scientists called Measles and Stahl, who grew E. coli in isotope of nitrogen N15. So normally it's N14. And what they did was they grew bacteria in this N15. So over several generations, you would have all the DNA containing nitrogen 15 as shown here. Now, what happened then was they took it away, replaced the nitrogen with N14, so any new DNA strands would be made of nitrogen 14. So what happened was they started off with DNA that was just N15. Generation 1, there was two strands of DNA created from one, and sorry, two molecules, I should say, of DNA created from one. One of the strands being new, one of the strands being old in both of them. Then the generation 2, those two DNA strands, DNA molecules went up to 4 DNA molecules. And in those 4 DNA molecules, you had the 2 original strands from generation 0 of N15, but all the others were N14. And it goes on and on and on. So basically, as you continued, each time the N15 numbers deteriorated in terms of the less percentage of the whole. You can see that in centrifugation. So the first generation 0, 100% would be N15. And then generation 1, it went to a mixture, so halfway between. Generation 2, you had and more amount on N14, and as you went over time, more and more N14 became found in the DNA. So how does DNA replicate? Well, and what are the key stages? Well, we've already gone through that with the one slide, but let's just have another quick look. The DNA must be copied in interphase. The hydrogen bonds have to unzip using helicase. The 3 nucleotides attach, and that is using the complementary base pair ruling. The DNA polymerase joins the new strands together. So just to re-emphasize that, this is a new strand here. So the DNA polymerase will attach the bonds here, 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 and so on to make the new strand of DNA. And then the DNA polymerase seals that backbone. And eventually you end up with 2 new molecules, half of the original and half. So how can we investigate and study DNA? Well, we can use polymerase chain reaction and electron. Polymerase chain reaction is much more familiar with people these days because it was used in COVID a lot to test for COVID, to take a small sample of DNA and increase it, magnify it to test for COVID. So artificial DNA replication is what this is. We can make tiny amounts become large amounts. It's vital in forensics, and as you'll know from COVID, it was used in testing for illnesses. DNA can be amplified through genetic processing. The whole process relies on 2 things. DNA made of antiparallel backbones. It's made with strands of 5' and 3' ends, and it only grows from the 3' end using complementary. It's different from natural replication because natural replication will code huge amount, like the whole DNA strands, whereas this only uses a small amount of DNA. We have to add something called a primer to start the process, and it is a cycle of heating and cooling to control enzymes as opposed to just continue. So let's have a look at the basic process. So we start here at number 1. We have a double sample stranded DNA sample. We heat that up to about 95 degrees C, which causes the hydrogen bonds to break and the 2 samples separate. We then add something called a primer. So this, you can see, is the primer here. And the reason we add a primer is because DNA polymerase, the enzyme that's going to attach all the remaining bases, needs a double strand to start on. So the primer we use using the RNA, we attach it on there, then the DNA polymerase is able to then work down the strand and create the rest of the DNA strand. This uses temperatures of about 55 degrees, which allows the primer to attach, which is called annealing. And number 4, the temperature is then raised to 72 degrees, which is the optimum temperature of the DNA polymerase that we're going to be using in this experiment. 72 degrees, well, the reason is that with the DNA polymerase we use is called Taq DNA polymerase, and that is an enzyme that's been isolated from thermophilic bacteria that are found in hot springs, so they work at a high temperature. allows us to control all the stages of the reactions. So if you're asked to describe PCR, these are the stages. Number one, the DNA sample is mixed with free nucleotides and DNA polymerase. Number two, it is heated to 95 degrees, breaking the hydrogen bonds, holding the complementary strands together, giving you two complementary single strands of DNA. Number three, the primers are added, which are short lumps of DNA, and the temperature reduced to 55 degrees, so they will attach and anneal to complementary base pairs, forming a short double-stranded section at either end of the sample. DNA polymerase can then bind to these short double-stranded sections, and you can then raise the temperature to 72 degrees, which is optimum for the Taq DNA polymerase, and it will add complementary free DNA strands. The polymerase will then reach down to the end of the strand of the molecule, and you will have your new molecules. And this process can be repeated and repeated at an exponential level. Now, if we want to separate DNA molecules, so let's say we've got a sample of DNA and we want to separate them, we can use electrophoresis to do this. So electrophoresis is used to separate DNA samples according to their size, and it can be used to an accuracy of one base pair in difference. It's widely used in gene technology to separate DNA fragments and identify DNA samples, and it uses something called a gel plate and a slab containing agar-rose solution. And this is basically a very impurity-free solution, which means that the agar doesn't block the movement. So how does this work? Well, at one end we have wells dug into it. So we have electrodes at either end of the sample. Okay, so one will have a positive charge, one will have a negative charge. The DNA is treated with an enzyme to cut it up, and then it's placed in a well at the negative end. And this is because DNA has a positive charge. So it will move away from the negative end towards the positive end. It will be immersed in buffer solution, and it will start to move towards the DNA is negatively charged, I should have said, so it will move towards the positive end. So the DNA negatively charged is put in the negative end, repelled from the negative end, and move towards the positive electrode. Shorter lengths of DNA basically have less resistance, they get less caught up in the gel, and therefore they move further along the gel. And the 6 positions can be identified by staining it with a gel, and you can work out where your DNA molecules are. We can then use DNA probes, and DNA probes are short, single-stranded bits of DNA that are complementary to a section we want to investigate. So let's say you had wanted to check if someone has a disease, like a genetic disorder. You could make a DNA probe that is complementary to that genetic disorder, and if that attaches, then you know that you've got the DNA for that genetic disorder. A probe can be labelled in 2 ways. It can be used for radioactive isotopes, normally phosphoryl groups are made radioactive, or it can be using fluorescent, which will emit a colour when exposed to UV light. Copies of the probe can then be added to the DNA because both are single-stranded. They'll bind to complementary base pairs through annealing. And it's useful to locate a desired gene if you're looking for one for genetic engineering, identify similar genes when you're comparing genomes for classification, and can be identified the presence of a particular allele which could increase your risk of a particular. PCR has been used for COVID testing. So these rapid production of large amounts of DNA meant that we could take a small sample of DNA from a human, extract, in this case, RNA from if the virus was present, and amplify it through PCR to identify if the virus was present. Advantages of this: it was sensitive to 1 molecule of viral RNA, and it could be amplified to 35 billion DNA molecules. It was very specific, as primers are designed to test for that particular viral. Obviously, it required equipment and materials, and at the start of the COVID pandemic, we didn't have these ready, and the results were not available for up to 2 days later because there was time for the PCR to be done. PCR can also be used for paternity testing, and here they focus on what we call short tandem repeats. And short tandem repeats are sequences of 2 to 7 bases that are repeated in your DNA sequence at particular points called loci on your DNA. And in the humans, 13 areas are used, and it is unlikely that anyone would have an identical sequence in any human. So we can use this to identify parental issues, or might use to test because a man claims that he's not the father to avoid financial support. Women might have had multiple partners and need to identify the father, and a child might wish to prove that the deceased person was their parents for financial reasons, or it could also be used for issues like if someone died in war and they need to identify the body. So just the HL students, then we need to know a little bit more about DNA replication. Replication relies on prime ends, and there is the idea of a 5' end of the molecule and a 3' end of the molecule based on the carbon numbers which are facing whichever way. And that is because this DNA strand is anti-parallel, so it goes in opposite directions. The enzyme used to attach the 3 nucleotides together can only attach to the 5' end. So for example, it could attach to this end here or this end here of the DNA strand. And therefore it builds in opposite directions. So it's going to move down this way. So my pen's not working. I put it down this way from this strand, up this way from this strand. And we can see this in the DNA replication process. When DNA is replicated, there are 2 strands. There is something called the leading strand, and there is something called the lagging strand. The leading strand is attached to the 3' end of the leading strand, and the DNA replication is continuous, whereas the lagging strand is made in smaller fragments, and that is because it's unzipping still here, and therefore you can't do it continuous from here and go all the way along. It has to be done in small segments as the DNA becomes available to have 3 nucleotides attached. Continuous variation. We need to know about a couple of different enzymes that are involved in the process of DNA replication for higher level. So the first one is DNA primase, which might seem strange that I'm about to say. That is actually a type of RNA polymerase. And what it does is it makes short sections of RNA strands that can then attach to the DNA template, so polymerase 3 can attach and start to create the new complementary strand of DNA. So effectively what we do is we have to add a double strand on to allow the DNA polymerase enzyme to attach. So we need 1 RNA primer to attach on the leading strand, but because we're making multiple strands on the lagging strand, you need regularly attaching the primase there. DNA polymerase 3 is the principal polymerase used to make new DNA. It binds to the 3' end of the template strand and builds a new strand, adding complementary bases up from a 5' to a 3' end in the new strand being built. It also proofreads each nucleotide, checking for errors in the DNA. Okay, so DNA polymerase 1 has 2 jobs as an exonuclease, which means it breaks down bonds in nucleotides, but it also has a job of a polymerase linking nucleotides together. Its job is it will move along the primer, replacing the RNA nucleotides with DNA ones. So it replaces the primer that starts off the strand with DNA ones. As it moves along, it can't move from along the lagging phase from Okazaki fragment to Okazaki fragment. So it will fall off in the gap, and then the next enzyme then joins those gaps together, which is called DNA ligase. So effectively, we then have one more enzyme called DNA ligase, and the DNA ligase will then fix those gaps between the Okazaki fragments in the lagging phase here. So these 3 enzymes are quite confusing. You do need to know the difference between them. So on the book, it's page 568 to 569. but here's a table for you which gives you some of the key factors for it. So I'll put above for what each function is, and on that obviously you need to know DNA ligase and DNA primase.